The story of Crispr-Cas9. Over the course of this video, I am going to attempt to explain to you the role of the Crispr-Cas9 system in the bacterial immune system and how its discovery has led to a breakthrough in modern genetic engineering. But before that, let's go back to basics. What do I mean when I say Crispr-Cas9, and how is it involved in the immune system of a bacteria? It has long been known that a living organism requires some kind of immune system, as survival is a constant arms race between organisms and pathogens who want to attack them. An ability to form some kind of immune response is vital. Without an immune system, an organism is left completely vulnerable. As humans, we have a highly developed immune system that allows us to fend off all manner of attackers. In multicellular complex organisms, like us, there tends to be what is known as an adaptive immunity. This is an incredibly complicated process involving all manner of immune cells and various other molecules interacting with the foreign invader and each other, eventually forming a memory of the attack via the formation of memory cells, which specialize in recognizing molecules associated with foreign invaders. This means that when a pathogen attacks for the second time, your body is ready and poised to fight back. But, like I said, this is an immensely complicated business that requires an immense amount of coordination and regulation. Surely there is no way that a single-celled bacterium could possibly possess an adaptive immune system, right? As recently as 10 to 15 years ago, scientists may have laughed you out of the laboratory for even suggesting that bacteria have an immunological memory. But astonishingly, it does appear to be true. Though the memory system employed by bacteria is very different from our adaptive immune response, the effect is remarkably similar. But how does it work? And when are you going to explain what you mean by the silly words at the beginning? Crispr and Cas9 are the toolkit at the bacteria's disposal in order to build immunological memory. To have a closer look, let's look at the bacterial genome. The bacteria's genome, which is simply the genetic material of an organism, like ours, is made up of DNA. However, whereas our DNA is bound into chromosomes and enclosed in a nucleus, the bacteria's genome is generally one circular chromosome containing all the essential genes for survival. In addition, bacteria have what are known as plasmids, which are small circular pieces of DNA containing useful, if not essential, genes. These can be passed and swapped between each other, and are one of the key factors in antibiotic resistance. But that's another story for another video. So DNA is the genetic code which allows for the production of proteins and all kinds of other interesting stuff that makes you, you. However, there is a sort of middleman in this process, RNA. DNA gets copied into RNA, and then this RNA is what is used to make proteins. It can also bind to DNA in the same way that DNA binds to its complementary strand. It is important to understand at least a little bit about RNA if I am going to explain Crispr-Cas9. Let's look at it this way. Imagine DNA as a kind of recipe book. It contains all the information for any kind of meal you want to cook. Let's say though you don't want the whole book, you only want to make one meal. One strategy would be to photocopy the page with the recipe you want. Then you can take this page into the kitchen and cook up a storm. If DNA is the entire book, then RNAs are a bit like these photocopies that instead of having the instructions for a meal, has the instructions to make a protein, or maybe a nifty CRISPR RNA that I'm going to talk about in just a minute. Bacterial genomes have been studied for some time. However, sometime in the late 1980s, a curious discovery was made. Short stretches of repeated bacterial DNA was found interspersed with random sequences, known then as spacers. Surely these served some kind of purpose. They must be doing something. No one knew. It would take almost 20 years before the mystery would be solved. In the mid-2000s, scientists started to notice similarities between these random spaces and viral DNA. This led to the notion that viral DNA was being incorporated into the bacterial genome. But if this was the case, why were bacteria doing this, and how? Enter these two bright sparks, Jennifer Doudna and Emmanuel Charpentier. Doudna was working at the University of California, Berkeley, and Charpentier was based in a lab in Sweden, when they decided to collaborate with one another to answer this question. They discovered that these sequences, known as spacers, were not only sandwiched between repeat sequences, but they were also found close to what are known as the Cas genes, one of which is the famous Cas9. This discovery would lead them to really understand what was happening. So here goes. When a virus comes along and infects a bacterial cell, it will inject its DNA through the cell membrane and into the cell. This is what viruses do. They hijack cells and use their machinery to replicate themselves. Once they've replicated enough times, they burst out. Suffice to say, this is bad news for a cell. In response to this, bacteria have developed a cunning defense system. When DNA gets into the cell, with the help of some of these Cas proteins, a certain amount is copied and placed in between these repeat sequences. This means that the bacterium, as long as it survives the initial attack, now has some of the virus's genetic material in its own genome. When the same strain of virus attacks again, an RNA strand is made from the DNA sequence containing these spacers from viral DNA, as well as these repeat sequences. After this, some other Cas proteins come along and process this RNA strand into what are known as Crispr RNA. These bind to another type of Cas proteins, and together they target viral DNA. To review, there are three types of Cas proteins. Type 1, which help insert the viral DNA into the bacterial genome. Type 2, which help convert RNA made from spacers and repeat sequences into Crispr RNA. And Type 3, which bind with Crispr RNA and help destroy the invading viral DNA. Don't forget that the viral DNA and the Crispr RNA are complementary. Using this system, the bacteria has a sort of arsenal against viral attack, allowing the bacteria to use the virus's own DNA against them. This is a fiendish trick. You may well be asking, this is all well and good, but how is this involved in genetic engineering? This is where the specific protein Cas9 comes in. At the beginning of their research, Charpentier was working with her colleagues in Sweden on a bacteria named Streptococcus pyogenes, a bacteria that is responsible for some nasty infections in humans. They realized that CRISPR-associated protein 9, Cas9 for short, was an integral part of the CRISPR system of this bacteria. Though they knew it was important, they didn't know what it did exactly. So they and their colleagues in both labs started working on Cas9's function. It turned out to be the third type of Cas protein. These are nucleases which cleave DNA at specific points, at a sequence that matches a CRISPR RNA. Let's think about this for a second. They found the protein that cleaved DNA at specific points, decided by the sequence of an RNA strand. This was when they knew they were onto something important. All they would have to do is make RNA molecules with the sequence that they would want to cut out of the genome and let Cas9 do its thing. Finding effective ways of making precise changes to the genome was the holy grail of biomedical research, and here it was, hiding in Streptococcus pyogenes. There was a step in the CRISPR system that I have left out. This is tracer RNA. Remember I said that the CRISPR RNA binds to Cas9? Well, for this to take place, the CRISPR RNA first needs to bind to tracer RNA. Once these have bound, it can recruit Cas9. Think of this tracer RNA as the mobile phone that Crispr RNA can use to call in Cas9. It just so happens in bacteria that the evolution of Crispr has made this a two-step process. But if you were to design this yourself, surely it would be easier to have a Crispr molecule with a mobile phone already present. This is what they did. They simplified the process by producing one long RNA molecule containing both Crispr and tracer elements. They called this the single guide RNA. The next step was to prove that it worked. They used a singular piece of DNA to test the cutting ability of Cas9. In the experiment, the DNA would be cut at specific points using a single guide RNA, and then the fragments would be measured. Using the sequence of this guide RNA, the length of the DNA fragments could be predetermined. The fragments at the end of the experiment were the exact size that was expecting, proving that it had worked. From this point, it went from a fascinating discovery for biologists into the creation of a new breakthrough technology. Through the work of Doudna, Charpentier, and others who worked alongside them, scientists now had a reliable, fast, and relatively inexpensive way of cutting and making changes to DNA sequences. This system is now being used all over the world for biomedical research and will no doubt aid researchers to make breakthroughs in treatment for genetic disease. One of the possible applications is something called genome editing. Certain genetic diseases are caused by mutations in the genetic sequence, leading to faulty proteins. An example of a genetic disease is Huntington's disease. This affects the nerves in the brain, causing a lack of muscle coordination and eventually leading to mental decline. The CRISPR-Cas9 system could be a potential way of treating such a disease. Theoretically, it could create double-strand breaks in the genome at sequences decided by the single guide RNA, which could cut certain mutations out of the genome. After this, the mutated sequence could be replaced by donor DNA, which does not carry the mutation, and this would allow a functional protein to be made. This is just one possible application of many for the CRISPR-Cas9 system. At the time, they did not know that they would be inventing a new technology. This breakthrough was simply born from curiosity into the genetics of bacteria. This happens a lot in science. However, there is another reason why I think this breakthrough is truly important. There is often this notion that science is something that was done by old dead guys, and is something that is basically over. All the good stuff has been discovered. This couldn't be further from the truth. Much of the research I've just mentioned took place probably whilst you were in high school. This stuff is essentially brand new. What's more, you can be sure that the next big development is being researched into as I speak. During your lifetime, there will probably be discoveries made that we couldn't possibly imagine in our wildest dreams. You could be doing that research. Science belongs to everyone. One of the great things about science is it doesn't matter where you're from, what your ethnic background is, or your gender. Science isn't about learning facts from GCSE textbooks. Whether it's biology, chemistry, physics, or any other science, it's about asking questions and making discoveries about our world and, hopefully in the process, improving the lives of everyone. If you're watching, get involved. Thanks for watching.